this is where we left off last time, and sorry I got cut off. I lost track of time. So just to go through quickly, remember we talked about mitosis. They line up in the middle, they split in half, and each new cell gets half, and now you've got two new cells with the same amount of chromosomes. The difference between meiosis and mitosis is meiosis actually has two divisions, and there's no doubling right here, okay? So instead of lining up in the middle, all of the chromosomes, they line up in pairs, and they're called tetrads. So when they split, a whole chromosome goes over here and a whole chromosome goes here. So it would be like both the chromosome ones are together and the twos and the threes and the fours. They don't line up in numerical order, you guys. Anyway, so then each chromosome goes. And then they do meiosis 2 looks a lot like mitosis. Then they split in half, and this half goes over there and that half goes over there. But now instead of ending up with one cell and getting two, in one cell and you get four. Okay. All right. And again, here it shows it. Here we have, the sister chromosomes, and they line up. And then so you can see now there's two chromosomes together. Okay. So when they split, the chromosomes don't split apart and half goes to each. This gets a chromosome and that gets a chromosome. And then the next one looks a lot like mitosis. They line up in the middle and they break in half in each half. The only difference here is there's only 23 chromosomes, whereas in mitosis there'd be 46. Okay, so you're going to end up with two daughter cells and then two more daughter cells. Okay, and the new daughter cells are going to have half the number of chromosomes, and these are your gametes. Okay, and you can see they have their prophase, metaphase, anaphase, telophase, and cytokinesis, and prophase, metaphase, anaphase. So you have 1 and 2. This one looks a lot like mitosis. This one's different because, like I said, instead of lining up in the middle with just single file, now they line up in pairs. So each new one gets half, they get the chromosomes. Each one gets one of the chromosomes from each. So one is going to be like when your sex cells divide, your chromosomes line up. Remember, you got your chromosomes half from mom and half from dad. Well, now the same thing's going to happen to one chromosome, whether you got it from mom or dad. And that's why we have genetic diversity. You never know which one's going to go. That's why, unless you're a twin, no two people have identical DNA because which chromosome got separated. Okay. So the division in meiosis 1 is 4 phases, and we just said that. The same as mitosis, but because there's two of them, it's 1, 2, 3. 1, 1, 1, 1, prophase, metaphase, anaphase, telophase. And then remember, cytokinesis is the cytoplasm and all the organelles dividing up. Okay. And then if you're talking about prophase 1 now, again, now you're going to see the chromosomes. So it's in that part, it's not going to be that much different. Okay. The chromosome pairs with their homologue, in other words, the chromosome, the other chromosome. So if it's chromosome 1, its partner is going to line up with it. Okay. Sometimes they cross over, parts of them cross over. So you even get more genetic diversity. So you might get a chromosome that you got from your mother, but a piece of it, one of the legs maybe, crossed over and you got that part from your dad. So now you get new chromosomes with different DNA. So you've got all this. And after this unit, we're going to start talking about genetics. So we'll get much more into that crossing over stuff. Okay. Metaphase 1, again, the difference here, they're going to line up in the middle. That part is the same. But instead of lining up single file, they're going to line up in pairs. So when they split, a pair of each is going to go to the sides. Okay. And again, you're still going to get the microtubules and look like it's pulling them apart. They're going to go to, but when they have the pairs of chromosomes together, they call those tetrads. Tetrad meaning 4. Well, you're going to have 4 parts. Okay. And then again, the microtubule is going to attach. Now, once they're lined up and you got the microtubules, then it's time to divide. Then anaphase 1, and again, instead of getting the chromosome splitting in half in each half, because in a chromosome, each half is the same. Okay, that's when they double, that's what happens. But in this case, they each get a chromosome. Instead of the chromosome splitting, they stay together and a whole chromosome goes on each side. Okay, so one chromosome from each pair moves toward the opposite poles. And then the sister chromatids remain attached. So those new chromosomes that have gone to each side, they still have the same information on each side. Okay, and again, they attach at the centromere, that's the middle part, or the end by the poles. And then they separate into Instead of the chromosome breaking in half, basically, and half going to each, you have the whole chromosome. And then you've got telophase, which is now where you're going to get, it splits into two. You get the cleavage and then you end up with the two parts. And then, of course, they're going to split the cytoplasm and the organelles and all that. So now you're going to end up with two new cells, but they have the same amount of chromosome material, but the chromosomes are not the same anymore. Okay? And cytokinesis occurs pretty much at the same time as telophase. While that cleavage is forming, you're splitting up the cytoplasm and all that good stuff. So now you're going to have your daughter cells, okay? But now those daughter cells are going to go through another division. Okay? So in animal cells, you've got the cleavage, the furrow. In plants, you got one more step, remember? You've got the plant cell. But now normally that would have been, if that was mitosis, that would have been cell division. Those cells would grow and go through interphase and do their thing. And then they double up their genetic material and they go through it again. But now in a sex cell, what happens is they don't double up their chromosome material. Okay. Now they're going to do meiosis 2, which looks a lot like mitosis, only now it's just half the number of chromosomes. Instead of 46 chromosomes lining up and splitting, now you'll have 23 splitting. Okay. So the chromosomes have already duplicated. That happened before meiosis 1 happened. So now, and it won't do it again. And that's how you end up with half the number of chromosomes. Okay. And again, here's prophase 1. The big difference again, and it's hard to tell here, but you have a blue chromosome and a red chromosome. Let's say these are both the number 1s, these are both the number 2s, these are both number 3s or whatever. And then when they split, the whole chromosome goes. So they line up, they don't line up single file like they did before. Okay. So that's meiosis 1. And then when you get into mitosis 2, you still have the 4 phases. And these, like I said, are going to look like regular mitosis. Okay, they're going to line up in the middle. They're going to split and follow those spindle fiber paths. And you're going to end up, with 2 new cells. And they're going to have, from one, you're going to have the same genetic information. And then you're going to have 2 more that are going to have their same genetic information. But if you look at all 4 of them, they're not the same as far as their genetic information. But again, we have prophase, metaphase, anaphase, telophase, and then cytokinesis. They just are 2. Okay. And again, like I said, meiosis, the second meiosis division is very similar to mitosis. The bigger difference is instead of 46 chromosomes lining up in the middle, 23 line up in the middle because they got half. Okay. And again, if you want to go through them specifically, prophase 2, you get your spindle former. This is prophase. You can tell it's in prophase because you're just starting to see the chromosomes. Okay. They don't look like a normal cell. You can actually see the little X's and they're kind of moving toward the middle, not there yet. When you get to metaphase, and again, metaphase is easy because it's when they're in the middle. But now they're going to line up single file. When they did meiosis 1, they didn't. They lined up in pairs. Okay? They lined up in their pairs of chromosomes, but they won't do that now because actually they don't have a pair anymore. But they'll just line up in the middle, single file, and then you get some crossing over. Okay? That happens. And that's why you get different genetic material. But again, like I said, we'll talk about that more later. But anyway, okay, so you got the crossing over, you got your meiosis 1 and meiosis 2. They go through all 4 phases. It's just the big difference is meiosis 1, you've got the chromosomes lining up together. And they call those tetrads because there's 4 of them, 4 chromatids. Okay, and again, the kinetophores of the sister chromatids attach to the microtubules and anaphase, then they separate. Okay, they're like an X here, and then they just pull apart. Okay. But again, like I said, in meiosis 1, they lined up in pairs of chromosomes. So a chromosome goes each side. That it doesn't split like it does in meiosis 2 or just mitosis. So the sister chromatids are from each chromosome now are going to go to opposite ends. And then you've got your telophase where you get your cleavage and you get your new cells. And as soon as telophase doesn't happen usually until those chromosomes get to the opposite ends. Okay, once they get the opposite ends, it's like, or poles, I guess they're called. Once they get there, it's like, oh, it's time for us to finish our job, and that's to form 2 new cells. Okay, and then again, cytokinesis separates the cytoplasm. And now, like I said, with mitosis, you started with 1 and you ended up with 2 daughter cells that had the exact same genetic information. With meiosis, you start with 1 and you have 1 division and get 2, and then you have another division and get 4. And the big thing is between the 2 divisions, they do not replicate their DNA, and that's why you get half the number. And it's important. If we don't have half the number, no, there can't be any reproductive cells because you need 46 full chromosomes. And if you have more or less, you're going to have issues. And again, we'll talk more about that when we talk about genetics. Okay, so each daughter cell is genetically distinct from the others. Okay, and from their parent cell. And that's not the case with mitosis. Mitosis is really asexual reproduction because you start with one and you divide to two, and the two new ones are exactly like the one it came from. That's not the case with meiosis. You're going to have 4 new cells, and they're going to be different. Okay. All right. And again, if you look, this looks very similar to mitosis. They line up in the middle, they separate in halves, and then you get your haploid cells. But again, because you started out with 2 that had different, now you go to division when you're done. Instead of starting with 1 and ending with 2, you start with 1 and you end with 4. Okay. When you compare them, mitosis conserves the number of chromosomes. Okay. Your new babies, your new cells are going to have exactly the same chromosome number as the one that it came from. And they're genetically identical to the parent cell. And again, that happens in every body cell in your body. That's why all your DNA is the same in every cell of your body except for your sex cells. Okay. Meiosis's job is to reduce the number of chromosomes. Okay. Because if we have the same number of chromosomes, when 2 sex cells come together, then we're going to have way too many chromosomes. So we take 2 diploid cells and you take 1 diploid and you divide and you get 4 haploid. And then when the 2 haploid come together, now you have a diploid cell again. And we're not like plants. We don't go through alternation of generation. We don't have a form of a haploid that becomes an individual. Okay. So again, that's the reason why we have meiosis, to get half the number of chromosomes. And again, you can see this. It shows you the differences between them. These pictures are in your book. You can go through them. And then you can see the whole picture and not me cutting one off. Sorry about that. Okay. So 3 events are unique to meiosis, and all 3 occur in meiosis 1. Okay. So again, meiosis 2 is very similar to mitosis. We can get crossing over, and I already talked about that a little bit, and we'll talk more about that when we do genetics, but we can end up with a new chromosome that has different, like maybe, there's like 4 parts. You got the 2 top parts and the 2 bottom parts. Well, maybe one of the bottom leg part gets, when the 4 of them are together, maybe they get kind of tangled up and one part will join there. So now you'll end up with a new chromosome that's different than any chromosome your parent had. It's a combination, but it's in a different part than it used to be. Okay. And again, instead of getting, when they line up in the middle in metaphase, instead of getting all the chromosomes lining up single file, now you get homologous pairs, the same pairs next to each other. Okay. And then they separate the pairs, not the chromosome, into chromatids, but the whole chromosome goes to the other side. So those are all different than in mitosis. Okay. And then the chromatids cohesion, and in other words, they allow the sister chromatids to stay together. You don't want it to break up. You want it to stick together. So then when it goes through the next division, then it splits into chromatids. Okay. In mitosis, the cohesions cleave at the end of metaphase. So in other words, when they split into the chromatids, that's when they start to cleave, is there in mitosis. Well, that doesn't happen until meiosis 2 in metaphase. Okay. So again, the big difference between them, and it's important, is meiosis, you're going to end up with half the number of chromosomes. It's really important. So meiosis 1 is really different because you've got chromosomes lining up in pairs, and the whole chromosomes going to a different side, and then they split like regular, it looks like regular mitosis, only now it's only half the number of chromosomes. That's the difference between the two. Okay. So meiosis 1 is where you get that reduction. And again, it's important so that we can reproduce. Okay. So, and here we talk about genetic variation. I'm looking at, you know what? We're going to quit right here because we're just about at 15 and I don't want to do what I did last time and cut you off right in the middle. Okay. So that's meiosis. That's sex cell division. You're going to end up with, start with 1 and end up with 4. Whereas mitosis, you start with 1 and you end up with 2. And the 2 are exactly like the parent cell, whereas meiosis, that's not the case. You get genetic variation, and that's why we have variation in individuals. Nobody, unless you're an identical twin, has the same DNA because of these changes. Okay, so we'll quit here and then we'll pick it up later on next time on genetic variation.